And we will go through a demo exploring the visual coding two-photon dataset.
If we're going to use this demo for two purposes, we're going to show how to extract data from the visual coding two-photon dataset from the Allen Brain Observatory. And we're also going to use this to answer a scientific question. So there was a paper in 2011 by Marshall et al. That performed two-photon imaging of a number of different areas of the mouse cortex. And we will see whether or not the data that is in the Allen Brain Observatory, so a different data set, also exhibits consistent findings with those reported by Marshall et al. So Marshall et al. Recorded from many different areas of the mouse visual cortex, including the anterior lateral area AL, the rostrolateral area RL, and the anterior-medial area AM. And they found that these areas exhibited more orientation selectivity than direction selectivity. And if we look at their raw data, their raw conclusions, we can see that orientation selectivity in these areas is much more prevalent than direction selectivity. And we want to go and look in the Allen Brain Institute data to see if that's also the case. So here we are, we're going to look at some of the data from the visual coding two-photon dataset. And this live script contains many instructions and examples for getting into this data set. So for example, we first find all of the sessions that are of the type visual coding and that are optical physiology or OPHYS. And then we can find all the sessions that have the structures of interest. So area AL, RL, and AM. And we'll find experiments where GCamp6F was expressed with Cree. We'll find sessions that had a particular imaging depth and where eye tracking was used, and where both gratings and natural movies were used. And we'll do some experiments on the first session in this list. So we'll go ahead and run these scripts. So here is a maximum projection image of the experiment. And we will plot the locations of the ROIs in another image. And so you can see these are the locations of all of the regions of interest that were identified in that brain section. We can look at the fluorescent timestamps. We can look at the delta F over F traces from many neurons, as we can see. And we can also look at the skewness of these neurons. The skewness indicates cells that are exhibiting substantial variation of their activity from the mean. We can also look at the responses to different stimuli, such as drifting gratings, and we can correct the responses so that we can measure the response during blank stimulus presentations and examine the responses of these some 120 neurons to 40 different visual stimuli. So you can see that different neurons are responding strongly to different stimuli. We can look at the orientation and direction index values of these neurons. And here we see confirmation of what we saw in Marshall et al., that these areas have stronger orientation selectivity index values than direction selectivity index values. And we can go on to plot tuning curves from individual cells. So these are tuning curves plotted in direction space, where the direction indicates the direction of motion of the stimulus. Here we see a cell that transitions from having responses in this downward direction at a temporal frequency of 1 Hertz to an upward direction as the temporal frequency increases past 8 hertz to 15 hertz. We can also plot temporal frequency tuning curves, as we do here. And we can make interactive plots so that we can look at the tuning of any cell of interest. So here I will change the cell number, and we watch as the tuning curve is updated. We can also look at measures of alertness, such as pupil diameter. Here we're plotting the average response, the average delta F over F, along with the pupil area. So when the pupil dilates, it generally corresponds to times when the animal is more alert. And so here we see the pupil is dilating at various times during the recording. And we can see that there's a correlation between the pupil area and the response, the delta F over F that's measured on average, so that when the pupil area is large, the delta F over F is also large. We can explore responses to natural movies. So these cells were also shown movie strips as a function of time. And here we can see for many of these neurons that they responded to different parts of the movie. So there are some neurons that responded to this part of the movie at around 10 seconds. There are other neurons that were responding right at the beginning of the movie. Here's another group that responded at the beginning and some certain phases. And so here we can study the responses of these cells to these natural movie stimulation. And we can also see which neurons exhibited reliable responses and neurons that exhibited less reliable responses. So here is a neuron that responded very strongly to one presentation of the movie, but responded almost not at all to the other presentations of the movie. So this cell was a cell that was not very reliable, had a reliability index of 0.02. And this is just some examples of the types of data that one can pull out with the Brain Observatory Toolbox and the Allen Brain Institute datasets.